so you're aware, this will end up on YouTube. Okay, so hi everyone, and welcome to today's UVic Center for Biomedical Research Cafe Scientifique. I'm Dr. Leanne Swain. I'm an associate professor in the Division of Medical Sciences at the University of Victoria. So I'll be your host and question moderator tonight, and I'm just going to start by acknowledging with respect the Lekwungen peoples on whose traditional territory the University of Victoria stands, and the Songhees, Esquimalt, and Wasanich peoples whose historical relationships with the land continue to this day. So for those who are new to Cafe Scientifique, what we do is welcome scientific researchers affiliated with UVic to share their work with the community. Pre-COVID, this happened in person, but for the time being, we're doing this virtually, of course. So please bear with me a bit, as I'm relatively new to hosting Cafe Scientifique, so thanks in advance for your patience. So as I mentioned, we won't have any interruptions during the talk. So just please note down your questions and post them in the chat later, or you can post them in the chat as they come to you. And I'll be reading those out later to our speaker at the end of the presentation. And the talk will run for about 45 minutes. So I'm really excited and happy to present today's and to introduce today's speaker, who is Becky Hoff. And she completed BSc Biology in 1998 at the University of Alberta and has a long-standing career in academic research. Since 2018, Becky has been the manager for the Facility for Biosample Preparation with UVic's Center for Advanced Materials and Related Technology, aka CAMTech, that works with the Center for Biomedical Research. So yeah, and then I will remind you that when we're done, we'll all try to raise our virtual hands using the Blackboard tool if we can figure it out in appreciation of Becky's talk. So with that, I'm just going to pass it over to Becky to continue with tonight's Cafe Scientifique. And thank you so much, everyone, for joining us.
Thank you so much, Leanne. So can you see me as well as the slides?
Yes, I can see you and the slides.
Okay, good, fantastic, thank you. So, hi, and thank you for the introduction, Leanne. Thank you so much for the territorial acknowledgement. And it's a real pleasure to be able to share my story with you tonight. So if you've come to a Cafe Scientifique before, you've heard stories from people who are passionate about science and who are passionate about a scientific problem. Now, my story is that I'm passionate about helping people do great science. So I've done a lot of different things in my career, and that experience has given me a skill set to run a multi-user biosafety level 2 facility for people who are not trained in molecular biology. So that experience has also given me the understanding that you have to create a culture and a space to enable a community to grow. So here's my story. I grew up in Edmonton in Treaty 6 territory in Enoch Cree lands, and I went to the University of Alberta. I did a degree in molecular genetics, and I don't really know what that is either. But in last year, I was extremely privileged to do a research project with Dr. Greg Goss. Now, my undergraduate research experience was a little bit unusual in what I got to do. So we were looking for a sodium pump protein in salmonid red blood cells. So salmon can go from freshwater to saltwater and saltwater back to freshwater and not have their blood explode. And that's actually kind of incredible and remarkable and unique amongst fish. So in order to study this in the lab, I learned how to work with radioactive sodium-22. We filled fish red blood cells with radioactive sodium-22. I learned to work with lethal levels of rat and poison dart poison. And I learned how to do underwater fish surgery to implant a catheter into a fish aorta. And I timed out my talk, and it's not too long, so I actually have time to tell the story of Greg came to me and said, I'm going to teach you how to do fish surgery underwater. And I thought, I have to be underwater, but no, it was actually the fish were underwater. And we're going to give you you're going to make a solution of bobane and warfarin, which are both rat poisons, and 3-tree curare, which is the poison dart poison. And we're going to put it in DMSO, which will allow it to go right through skin and membranes and straight into the cells. And we're also going to fill them full of radioactive sodium-22. And I kind of looked sideways at him, and he goes, "It is incredibly dangerous. And you will either be the safest scientist or you will be dead." And I love the old days sometimes. I turned out I was a safe scientist. And it was an incredible experience, and the skills that I got were unique and inspiring. It made me love bench science. So it really set me up for the rest of my career. So after graduation, I moved to California, to Kumeyaay lands, and lived in the city of San Diego. I had the incredible luck to land a job at a mid-sized biotech company called Vical. At Vical, I worked on a lot of different projects, ran disease models of melanoma, prostate cancer, breast cancer. Oh, sorry, turns out my slides aren't advancing. Thank you, technical support in the Hoff House. So this is a picture of the fish, and then I got to move to San Diego, which is stunningly beautiful. And so at Vical, I got to work on a bunch of different cancer models. So I worked on glioma, breast cancer, ovarian cancer, prostate cancer. I primarily worked on melanoma, and on melanoma, I got to work on a product called Alovectin 7, which at the time was the most advanced therapeutic for malignant melanoma in phase 3 clinical trials. So that's like a real, it's sort of a dream of every cancer biologist, every animal model person to get to work on something so important. I also got to work on a malaria vaccine there, and my pet project was working on better formulations and delivery methods for multiple sclerosis therapeutics. After California, I spent a few years in Switzerland eating delicious chocolate and hiking up beautiful mountains. And then in 2005, I moved back to Canada and I settled in Victoria. In Victoria, I've done a few different things. One of them was doing data quality control for a chemistry company. I ran a home-based childcare center for a while. And I worked for Dr. Caroline Cameron doing syphilis research for about 10 years. I hope some of you, a few years ago, she did a Cafe Scientifique, and I hope you got to see it. She is a remarkable scientist and an amazing person. So there we go. Fix these slides. In 2018, I got the chance to run the FBS. So the FBS is the Facility for Biosample Preparation. It's an open access, multi-user molecular biology facility created by CamTech. CamTech is the Center for Advanced Materials and Related Technologies, and that is all a mouthful. So in a nutshell, CamTech is a collection of physicists and engineers and chemists who really care about material science. And the FBS is what we would call a core facility. In other places, they would call it an incubator or a full-service co-working space. So why do we need a space like the FBS? Well, it's because of how modern science is done. So in the last 100 years or so, science has been done in a relatively siloed way. As a scientist, you're an absolute expert in your subject. You get to know as much about it as possible. You really delve deep. And this approach to science has been a staggering success. We've been able to solve some problems in the world with it that have revolutionized the world. So one example is Carl Djerassi and his team of chemists. They single-mindedly focused on synthesizing and formulating a birth control pill, and it took them about a decade, but that single-minded focus on that sole goal really, I mean, the birth control pill has revolutionized society. So as scientists have delved deeper and fully explored their areas, and they've developed and honed their tools, what we've started to understand as scientists is that the tools and the approaches and the expertise you have in one field can be used to work on and solve some intractable problems in other fields of science. So more and more and more, the idea of interdisciplinary science is becoming important. So there are a few ways that you can do research in a field that you are not an expert in. And the first way is to find a collaborator. So if you were a passionate scientist, you might find another passionate scientist in the exact area that complements your work. And so when this happens, it is ideal. This is the best of all worlds. But it is really hard to find someone who is as excited about the exact same problem as you and who has the exact expertise that matches your needs. So it's really hard. Another way to do research in an area that you're not an expert is just to pay somebody to run an experiment for you. So one problem with this is that you can't always find somebody or a place that will do or that can do that type of experiment. Another problem with fee-for-service or contracting a scientific experiment out is that you have to decide ahead of time exactly how you want the experiment run. And you get that experiment run in that experiment in that exact way. But you're not able to change the experiment on the fly in response to new information and new data coming out of that experiment. So both of those approaches are good. They have their problems, but there's also one really important problem with both of those approaches, and it's that it is slow. You send your sample away, and it can be months and sometimes a year before you get data back, and while you're waiting for that data to come back, you're not able to move forward with that project. And so it's either not an iterative process or it's a very slow iterative process. So that's where a facility like the FBS comes in. And now I'm going to tell you about one of my favorite research groups at UVic. And just as an aside, my family tells me that I am literally the worst at superlatives. But the truth is, each of my daughters is actually my favorite daughter. So I can be the worst at superlatives. And Professor Heather Buckley in the Department of Civil Engineering works with a research group of chemists and engineers. They are absolute experts in green technologies and clean, safe water. And they sought to apply the these areas of expertise to the problem of biofilms. So there's Buckley Labs at Green Safe Water. And so what are biofilms? Biofilms are when a bacteria or other microorganism adheres to a surface and they change and they start to grow and they get really sticky and they can be really hard to kill and really hard to remove. And you can imagine that this would be a problem for something like a surgically implanted device, if you got a biofilm on it, or say if you got a biofilm formation on water treatment pipes. So this would be a real problem for both individuals, but also for communities. Now, there was this lab, the Buckley Lab is a lab full of chemists and engineers, and they're fantastic at what they do. They are not microbiologists. And so they have this plan to study biofilms, and they have the confidence to take on this new challenge. But there's a lot of specialized equipment, techniques, and regulatory hurdles or gatekeeping that is required to do this type of research, especially if the microbes that you're going to be studying are capable of causing harm or infecting humans or other say stock or livestock animals. So fortunately, there was a brilliant graduate student named Anna, and Anna had an equally brilliant undergraduate student named Matt, and they were keen to learn how to do microbiology. But first, they needed to know some absolute basics, like how to sterilely transfer water from one container to another. And they had to learn this because this isn't something that a civil engineer would normally have the opportunity to learn. So they needed training and expertise, but they also needed things like incubators, pipettes, microscopes. So Heather and Anna came to me, and it turns out that I have incubators, pipettes, and microscopes, and I also have a lot of experience with bacteria. Thank you to the Cameron Lab for that. So rather than have to start a microbiology lab from scratch, they were able to start doing microbiology pretty quickly. I taught them how to use the instruments and the basic techniques of microbiology, and they really took it from there. Me and Anna and Matt worked together to troubleshoot experiments, and in no time they were way more expert than me. They then moved to a type of bacteria that was more relevant to human disease and to water quality. So at this point, this is the point in their research where we have to start bringing in other parts of the university community. So at UVic, we have a biosafety officer that helps ensure that both the scientist and the community at large are safe when people are doing research on these potentially infectious organisms. Occupational health and safety also has a person in charge of making sure that hazardous waste is properly disposed of so that the community is safe from the waste created from these organisms. So there's a lot of people involved in facilitating this type of research. And where I come in is I liaise with and act as a go-between and an interpreter and a helper for the researchers, and and the safety people. So the other problem is you can't just go out and buy potentially infectious bacteria. There are regulations, and there needs to be regulations on this. And so the picture you've been looking at is Matt in the hood. So I love this picture. Thank you, Anna, for taking such a great picture. So you can't just go out and buy potentially infectious bacteria. At University of Victoria, we have science stores, and this is a picture of the team at science stores, and they help buy the materials and supplies needed for research. But they're particularly important for buying anything that would be considered biosafety level 2, because they're trained in making sure that the regulatory paperwork is all in place. They're also trained on the shipping and receiving of dangerous goods, and they are fantastic, and thank you so much to that team. So I'm going to show you a picture from Anna and Matt's research, and this is actually a picture of a biofilm in a small well. And the green stain on there is for live bacteria, and the red stain is for dead bacteria. And so we've taken pictures with a green fluorescence filter on the microscope and a red fluorescence filter on the microscope. And then we've overlaid the two pictures so that you can see how in the corners of the well, it's a little cylinder, the dead bacteria and the live bacteria sort of grow together and form a larger, thicker superstructure. And as you get more towards the center of the plate, you see these thin films of live bacteria. And I think this is just an incredibly beautiful picture. And I was so excited to see this type of image coming off of the microscope. It was very exciting for me, and anybody who's been in the lab with me knows how excited I get about microscopy. So my facility provides a place for the wider university research community to learn techniques, to have access to equipment. It also acts as a portal to access other university supports. I help researchers navigate the regulatory and safety aspects of doing research using biosafety level 2 organisms. And Anna has now graduated. Congratulations, Anna. And Matt has taken over the project fully. He's training new members of the Buckley Lab in the specialized techniques that they've developed. And importantly, he's also giving advice and support to other labs who are interested in doing biofilm research. So, and as an extension of that, some of the newly trained lab members are now collaborating with organic synthetic chemists to improve and expand on these projects. So what we have is engineers working with chemists, doing microbiology, and they're all putting these diverse skill sets together to solve some of our greater community problems. So far, the story has been pretty straightforward. A professor and lab team use an open access multi-user facility to open a door to new research possibilities. And the initial intention for this facility was exactly what happened with the Buckley Lab. An engineer needs a place to test new materials. Worked out perfectly. But something really interesting happened. Anna's friend and lab mate, Caitlin, was having trouble with a fiber optic sensor that she was making. She thought it might be a manufacturing problem. She was actually making the fiber optic sensors, and she thought it might be a manufacturing problem, because sometimes they would work and sometimes they wouldn't. And she wasn't really able to tell when a sensor would work and when it wasn't. And so she was trying to figure this out. And one day she was hanging around with Anna and at the microscope, and they realized that there was a microscope in the facility that could handle really strangely shaped objects. And so Caitlin and I started taking little movies of her, and here is Caitlin and Anna. So that's Anna in the forest, and Caitlin looking like she's already graduated, which she has. Congratulations, Caitlin, and congratulations, Anna. And we started taking these little movies that went through in a stacked way of her fiber optic cables. And if you can look, and I wonder if my cursor actually works here, I hope it does. If you can see, this is actually the center diameter of a very, very, very small fiber optic cable. And you can see how there's a hole down the middle and the end is cut, and the outside is actually coated in something really important and really special, and I really am not a chemist. And she was able to do quality control on these sensors before she went out into the field to test them, and it really helped speed up her research and make it way more effective and efficient. So I'm going to show you some of the other research. So having an instrument to check these sensors is really important, but the actual most important part of this story is that Anna and Caitlin, working on very different projects, Anna was working on biofilm, Caitlin was working on fiber optic sensors, they have a culture that encourages discussion and group problem solving. And that is how Caitlin was able to get one of her problems solved, because of this culture. So now I've shown you some really pretty pictures that the microscope can take. We can do all sorts of things in the facility, but microscope pictures just are the best things for a presentation like this. And we have some really beautiful ones. So the next set of pictures that you're going to see are from the Willerth lab, and they're biomedical engineers, and they do regenerative medicine and spinal cord repair. And I wonder if I can get that. There should be a few. Oh. So what you're looking at in this picture is fluorescently labeled neuronal cells and other types of cells that are used. The fluorescent dyes are used to identify different parts of the cells and different types of cells in a mixed cell. And when we layer the layers of fluorescent microscopy on top of each other, we get a lot of really amazing information from this. And this is a beautiful cell. So this big green one is a neuron, and these other ones are some of the other bits of the cells. And thank you so much, Megan, for just taking gorgeous pictures. So the next set of pictures is way less dramatic and a little bit less beautiful, unless you're a biofilm researcher, and then it's gorgeous. And what this is it's a study of bacterial films from the Hoare lab. And now the Hoare lab in chemistry uses lasers to look at the space between surfaces and something stuck to the surface. It's amazing research, and they're taking this incredible laser and microfluidics knowledge, and now they're starting to apply it to bacteria. And Tasha, the grad student, would grow some E. coli on a specialized quartz crystal, and they would do all of their studies, and then they would have to check and see how much bacteria was there. So the black and white picture up here is just a regular bright field picture of the biofilm, and those are all E. coli. And what she was using, there's some little red circles around each of the dots, and those little red circles are actually the computer program associated with the microscope counting each of those little bacteria, because it can take a really long time to count them. So that's the computer program counting those bacteria. And if you look down at the black and red one, she's also stained them. And this is a fluorescence image of those same bacteria. So we're looking at confluence and spacing. And it's a real workhorse way to do microscopy, but it's really effective and efficient. And there's laser people working in the facility. It's really, really cool. One of the other research groups is in the biology department, and they're salmon behavior experts. And the salmon behavior experts in the WANIS lab, they go out onto the water, into the rivers for months, looking at different ways that the environment stresses salmon, and they take samples of the salmon blood to look for indicators of stress, to actually measure the stress. And one of the indicators of stress is called cortisol. And they bring the blood back, and they need to test to actually see how much cortisol is in the salmon blood. And so they came to my facility and ran, you can see this 96 well plate with the little blue. They ran an assay that looked a lot like this. I didn't think to take a picture of their assay, so this is another plate. But what they did looks a lot like this. And they were able to actually quantify the biomarker of stress in these salmon based on their experimental design changes to the salmon's environment. And as you can probably guess, that particular bit of research was near and dear to my heart. If you remember back to my undergraduate research, doing salmon research. And if you look to the side, there's kind of a starfish shaped red picture on a black background. And what that is the Biomedical Engineering Lab Course 401 has 3D tissue printers, and they would program and print up these 3D shapes and bring them to my facility. Now they look sort of like clear blobs, but once we get them under the microscope and turn the fluorescence on, we get to see these beautiful 3D printed pictures of their shapes that they programmed. And it's really wonderful to get to work with students doing this sort of thing. And down in the bottom corner, we have a group of chemists who come in and use the facility. And full disclosure, it's Fraser Hoff's group. And yes, we are related, but it is only by marriage. And the Hoff group comes in, and they're organic chemists. And they make molecules. And one of the molecules they made was to study a protein that binds epigenetic markers. And epigenetics is a buzzword these days. So the epigenetic markers are linked to different cancer outcomes, particularly in this case, in this protein was prostate cancer. And so what happened in the facility for biosample preparation is the chemists started growing E. coli that expressed this human protein and then purifying out the E. coli. And if you look, the bottom picture down here, those are actually 4-liter Erlenmeyer flasks. They're really big, and inside each of them they grow 1.5 liters of E. coli. So this is like big scale protein production, and that started fairly early in the history of the FBS. And since then, that project has turned into a long-term project with AstraZeneca for prostate cancer therapeutics. And I'm really excited about that particular bit of research. It's a nice long-term outcome for something that could happen in the facility. So the best part of these very different groups coming in and working in a shared space is that they're all talking about their experiments. There's cross-fertilization, and the young scientists and the graduate students are seeing possibilities for new directions in research, but more importantly, they're starting to think that these areas that seem so foreign and unattainable are absolutely within their technical and physical reach. So up until this point, I primarily talked about academic research. The research community is actually much bigger than that, and especially for interdisciplinary research. Interdisciplinary research is done by all sorts of scientists, and if you think back and remember when I was talking about collaborations, well, sometimes what happens is that you have two scientists that perfectly complement each other, but they don't have a physical space to do their research. And this is where another group comes in. And that's a small but mighty startup company in Victoria called Spayvac for Wildlife. And Mark Fraker is a wildlife biologist. He has about 40 years of experience doing wildlife biology, a lot of it with feral horses. And David Kroger Dr. David Kroger is an immunologist and biochemist, and they got together and they contacted me through another university-based partner, and that's the Research and Partnerships and Knowledge and Mobilization. It's quite a long name, but essentially what this unit does is they help outside companies and groups access the university and do contracts with university researchers. They facilitate this. And so Lindsay over in our PKM got Mark in touch with me, and they really needed a space to do some basic research and a place to manufacture their vaccine. And what their vaccine is a birth control vaccine for feral horses and other types of feral populations. It's a humane way to help with population control in these feral populations. And so Mark and David and I met and we set things up, and now Spayvac has found a home in our facility. And they're now doing multiple trials all around the world. And their birth control vaccine is having a real impact, both ecologically and humanely, on these feral populations and the ecology where they live. If you're interested in learning about the problems of feral horses and other feral populations, you really should go visit Spayvac for Wildlife's website. It's very good. Now, a knock-on benefit to having Spayvac scientists working in the facility is that David has a wealth of experience and knowledge, and he is generous with his mentorship of students who are learning the tissue culture ropes and really stretching outside of their area of expertise. And he's been really helpful for a lot of the students coming through. There's several small and medium-sized startups and companies who do work in the facility. And along with the economic benefits that they provide, they're also creating jobs for UVic graduates. And they're providing mentorship to researchers in both the science part of it, but uniquely in the entrepreneurship side of it. They're really mentoring young science entrepreneurs to take their ideas and develop them. And so that's a really unique opportunity for a lot of students and postdocs and other researchers to have that sort of mentoring. So now we have interdisciplinary researchers in the UVic academic world and in the greater science research community. And there's a third group that is really vital to this story, and it's the wider community. I love to do outreach. I love to talk to the scientist in every person. And I've been lucky enough to work with Dr. Elaine Humphrey, and she manages the Large Electron Microscope at UVic. And her and I regularly do outreach to school groups and give tours to the general public of our facilities and science in general. And last spring, just before COVID hit, I got to do a really fantastic fluorescence microscopy project with my Girl Guide unit. And I know that you will all be very surprised to find out that my Girl Guides do a lot of STEM in their programming, but that's okay. Then that's science, engineering, materials, and technology, not in that order. And the project that I did with my Girl Guides was they first learned how to do scientific illustrations, and then they went out into nature and collected samples, and they observed and drew their samples. And then we brought them in and looked at them under the regular optical microscope. And then a lot of things in nature have autofluorescence. You don't always have to stain to see something under a fluorescent microscope. And it turns out one of our girls, and all of the girls were able to see something interesting under the fluorescent microscope, but one of my girls, Emma, she chose, and I really hope I can get this one to, I really want this to come in layers. I'm not really sure how I'm going to get that to happen. Emma picked up a squished worm, and the worm was squished. She didn't squish it, but when we got it under the microscope, we were able to see the different layers and structures in the worm. The worm just sort of looked like a squishy mess under normal bright field. But then when we put the fluorescence filters on, we were able to see all of these new and interesting ultrastructures inside of the worm. And this was fabulous and exciting, and the pictures are beautiful, as you can see. And we had a wonderful session doing this. The girls learned a lot. And they came in and they got to experience real hands-on science. But something more important happened in that experience. And it's that they got to see real scientists that looked like them doing real experiments in a working lab. So I would really like to thank a few people. I'd like to thank Allison, Zoe, Alok, Chelsea, Tong, B, Eric, Austin, and whoever else was around on those evenings when the girls came in, because they are an inspiration for this next generation of 10 to 12-year-old girls to become fearless researchers. So there are a few things, and I really would like this one to work. So I don't know if it's a little bit hard to see, and I'm so sorry. It was supposed to fade in, but technical difficulties. So the point of the talk was it takes a village to do research, and there are several aspects. There's 4 parts in order to create a thriving interdisciplinary research community. And the first is physical space. You need advanced instrumentation, you need infrastructure, you need space. And the second really important thing is support. You need somebody with technical skills, you need somebody to do training, you need support through the regulatory, important regulatory steps to doing that type of research. The third really important part is growing a culture. It's a culture of mentorship, of open discussions, of group problem solving, where you have people from different research backgrounds discussing problems and getting excited about each other's science and interested in bringing new and fresh ideas to the process. And it takes a real open culture to have the confidence that your problems will be well received and that you'll get the support you'll need from that. And when you have those three things in place, what needs to happen next is building connections. And that's among scientists and engineers, that's across campus, that's across disciplines, and most importantly, we have to build connections to the wider community. And that means you, everyone who's here tonight, is part of that, because the wider community comes and supports physical space for scientists to do this type of research. And they help create a culture where we have these types of interdisciplinary approaches and open approaches to greater societal problems. So the pivotal point in this, creating a research village, is you. So now you are part of the research village. Do you have any questions? And Leanne, would you mind opening that up? And I'm going to see if I can make this go down.
Raise our hands as a virtual clapping for such an amazing presentation. So I don't know if you can see that.
Thank you everybody for coming so much. This was such an honor and a privilege to be able to share this story with you. I'm absolutely gobsmacked that one was asked, and two, that you showed such an amazing interest in community spirit in this. So thank you everybody.
Wow, that's an amazing notification response. That's awesome. That was an amazing talk. Thank you so much, Becky. It looks like we have a question in the chat and some questions as well. So I'm going to start with a question from Jim and Betty Hesser. Is SPAVAC involved in the OP experiment to control deer populations?
I don't think David or Mark are here. They might be. I don't know. The stuff that I've been working on them with hasn't been, but they might have been. I can't say for sure. Go to their website. It's probably part of that. It's probably on there.
That's a good idea.
Yeah.
I have a question for you while we wait for other questions to come into the chat. So I don't know if everyone appreciates how difficult it is to just like adapt and learn new techniques. So you already knew a ton of different techniques, but imagine in your role, you're also just like, okay, here's a new research group and I'm going to have to learn a whole bunch of new techniques. What do you think are important characteristics for that? So for some of the people who want to appreciate from the general public, but also for some of the trainees who are tuned in to the talk, some of the key characteristics for just adapting and learning new techniques? You can hear my dog just like drinking his water in the background.
I would say I'm not a great scientist and I don't know anything, and I am super grateful anytime somebody is willing to share their experience with me. It's I think really coming into it with an openness is really important.
Yeah.
I think that's because at this point we're all pretty good at stuff. We can learn stuff, and we have good hands, and we have well-trained brains. But I think it's about attitude, and you come in with an openness and a willingness to go back to the beginning and learn from scratch. And as Anna and Matt showed, they went from 0 to hero in a few months. It was remarkable. They did it with an absolute willingness and openness.
Right.
To
so that willingness is
learn
still
it.
Yeah.
And they didn't think that they really came like, let's learn this.
Right. So you would say kind of be humble and just be open to learning from experts. Yeah.
Yeah. Learn from wherever you can. Everybody's got something interesting to say.
For sure. And I imagine that people look a lot to you for direction. And so how do you deal with that? Do you what do you think about when someone first comes to you and says how do I do this thing? And if you're like, I don't know how to do that.
I don't know, let's learn. Let's figure it out. I'm sure there's a way. Let's figure it out.
Yeah, totally. And then I'm sure you have your resourcefulness to connect with the right people. But I think you're also underselling yourself because you're amazing at a lot of things. Okay, so we have another question here. Do you ever have to collaborate with difficult scientists? If so, how do you handle that? Great question.
I have been pretty fortunate in my life to not have to collaborate with a lot of very difficult scientists. So I've been really lucky, really lucky. I think the one thing I would say is if you're having real communication issues, and that having really clear expectations of what you're going to do and what the other person is going to do, and having them written and verified by both people. That's been a technique that's really helped save me, especially when I'm not super clear. Like if I'm having trouble communicating what I'm expecting, actually having the conversation and then writing it down and saying, did I understand this correctly? That's been really effective. And sometimes there's just people who are hard to collaborate with because
Yep. That's a great approach. And yes, absolutely.
But write it down and make sure that you're both having the same conversation.
Yeah.
That's what I do.
How do you keep all these various and very diverse research projects and things going on in this space? How do you keep track of all that? And what's your strategy for kind of organization?
Camtech has a online booking system for accessing all of our facilities. And there's a manager for that and many things in Camtech. And Milton Wang is a technical marvel, and he has helped set up a system so that we can track training and safety and protocols, and it also allows people to book spaces and instruments. So there's the physical side of it, and Milton has just made that a dream. The scientific side of it is sometimes I'm good at that, and sometimes I'm less good. Honestly, I don't get to run my own research anymore. So I don't do research for me anymore. And I need to meet that. I need to meet that need in me to do bench science, because I've been a bench scientist, and I probably shouldn't say this, but now I think technically it's 24 and a half years I've been working on the bench. And I just get so excited about everybody's research. It's really fun. And it's really interesting And I have a black notebook that I write everything I do down every day in, because otherwise I would forget everything. Because I have a lab notebook that I write down what I do as I'm doing it, and conversations that I have, because otherwise I'll forget. And I also have a very complex sticky system. So the little colored stickies, if something needs to be done, I put a sticky on the day. You know what I mean? Yeah.
Okay, awesome. So we have 2 more questions. So I love this. Like, you have more time, a bit more time, because we started a bit late. So if you have more questions, just put them in the chat. So Gayla B says, loved your presentation. Has there been a demand for these projects that exceeds your capacity? And if so, what criteria do you use to select those or select projects?
So yeah, there are definitely projects that I don't have either the physical space for or the technical instrumentation for to be run in the facility, and that's just sort of like I'm sorry, I can't do that here. Maybe, and I try to help them find a place to do it. In terms of expertise, I'm just really upfront about what I do know how to do and what I don't, and what supports I can provide. The criteria that I use I don't filter or judge projects coming in. I also leave scheduling of projects unless it requires a lot of my personal time. The scientists who come in, I allow them to schedule their own time, and that's where the system that Milton has set up, the online booking system that Milton set up, they will come in and schedule their time. And so they manage a lot of their own scheduling, unless they need me personally to come in and help them, and then I work as much as I can with them. So far, knock on wood, I haven't had too many projects that we couldn't handle.
Good stuff.
Yeah.
Yeah. Okay, so we have another question from Natasha Milosevic, or Natalia Milosevic. Hey, Becky, really great talk. You're an incredible scientist and mentor. Really grateful for all your advice and help when I was at UVic. What are some new directions / goals / resources that you think would be useful for the center?
Oh, Natasha, did somebody ask you to ask me that question? Because in my dream of dreams, we would have some space to do more support for some of the other faculties, like the faculty of nursing, where we could maybe even have a place for patients to come in and get samples done, and we could maybe collaborate more with the Center on Aging. Well, that's my secret, I guess at this point, not so secret dream. But if I could imagine a center, it would have some space and facilities to help bridge the gap between certain types of medical and social science research and molecular biology research, and there's a space in there that I think that I would love to facilitate some of that stuff. That's my secret dream. But you know, I've got I think about another 20 years in my career, so we'll see what I can get done in that time.
Yeah, there could definitely be some outreach, and then there could be some dedicated kind of like outreach for students as well, like for your like Girl Guide groups and like other student groups, like that's
yeah, I'd really like to get more people in different types of communities able to access space to do this type of research.
Amazing. Awesome. Well, I think that's all we have for questions. Awesome questions, great participation, everyone. And just a wonderful, very dynamic and inspiring presentation. I'm super excited about the facility for biosample preparation. I think I'm saying that wrong. It's the FBI.
You're saying that right.
Okay. And I'm so excited for your work there. Seems like a really diverse and super interesting group of scientists that you get to work with, and so many cool projects. Like Heather Buckley, she says she expresses in the comments her thanks and that you're so awesome to work with. So sounds like you're just doing an amazing job, Becky.
Well, thank you. If you want, you could let Jeremy know that he's actually the director of the center.
All right, I'll pass that on. And thank you so much for your presentation. I think you inspired a lot of people, and super exciting that you're doing so much outreach, and all the girls that you inspired in the Girl Guides. Like, wow, that was an amazing microscopy picture. You were so awesome.
Thank you so much, and that really means a lot coming from you, Leanne, because
okay. All right. Thanks everyone for attending. That was a great Center for Biomedical Research, Cafe Scientifique. So lucky to have an awesome speaker, Becky, tonight. And thanks so much, everyone. Have a great week. Stay safe. And thank you so much, Becky.
And thank you to the Center for Biomedical Research for providing an opportunity to do this research. I am extremely grateful to you. And thank you, Leanne.
Yes, awesome. Have a great evening, everyone. Bye. Thanks, Becky.